Dr. Ulrike Mathesius is investigating how soil microbes shape the plant. She developed and applied techniques at a molecular, cellular, and whole plant level to define mechanisms that symbiotic and pathogenic organisms use to manipulate their environment. A central idea of her work is that microbes have hijacked plant signaling pathways for their own purposes. This has implications for utilizing microbes to alter crop plant performance and for trying to develop nitrogen-fixing symbioses in non-legumes. Dr. Mathesius, congratulations, and I invite you to present your lecture, Plants Control Microbes, Microbes Control Plants.
Thank you very much. Thank you. It's a big honor to get this award, and I want to thank all the people who supported me along the way and opened lots of doors for me to be here today. So I want to talk to you about a part of my research about signaling interactions between plants and microbes. But I want to start with a picture of a human. So this is a human with all its cells, obviously, but there's really a lot more to us than just human cells. Our cells are outnumbered by about 10 times by microbial cells inside us and around us. There are bacteria in our nose, in our throat, maybe in our lungs, certainly in our intestines. There might be nice biofilms forming in our teeth right now after the nice cakes outside, which are actually highly complex multicellular structures. And maybe there might be some parasites in our blood or elsewhere. And all these microbes have functions as well. They might help in immune regulation or in digestion and all sorts of other things. So how about plants? Well, it's the same for plants. Plants have evolved in surroundings with microbes since the start of their evolution. And here are a few examples. So bacteria colonize the inside of plants. Here's some nitrogen-fixing bacteria in sugarcane stems. There are bacteria that colonize root surfaces. These are mycorrhizal fungi that colonize the inside of roots and help with phosphorus nutrition, and bacteria inside nodules of legumes that fix nitrogen. So why do microbes live in and around roots? Well, basically the plant is a very good food source and also provides a lot of niches for bacteria to live and to hide and to evolve. It's estimated that maybe 5 to 30% of the carbon that's fixed during photosynthesis in the shoot goes straight to the root and is actually exuded into the soil. It goes away. But the plant doesn't really just lose it. It has specific functions. Quite a lot of it is exuded as sugars or organic acids, which are great food for bacteria. In return, the microbes that eat that food serve a lot of biological functions that help the plant. For example, in mineralization of nutrients, provision of nutrients, sometimes pathogen protection. Some microbes, of course, have taken advantage of this as well and harm the plant. But if you look at the microbes on roots or on plants, it's not just that randomly all the bacteria come along and eat this food. The colonization of plants by bacteria is quite specific. The species diversity on root surfaces is actually a lot lower than the species diversity in the soil, even though the concentration of microbes is a lot higher. You can see here a picture of root hairs colonized by microbes, and different genera of microbes are labeled with different colors. So you can see they are spatially in specific places, and there are also certain groups of microbes. In some cases, these interactions are really specific. So, for example, this is a nodule of a legume root colonized by nitrogen-fixing rhizobia that reside inside these nodules, and they actually fix nitrogen. So this is a process that's really important for converting atmospheric nitrogen into nitrogen that the plant can use. It's very important for agriculture. So these ones are really like little factories in which nitrogen fertilizer is produced for the plant. And it's a very specific interaction. The plant doesn't just let everyone in. It invites certain bacteria in and excludes other for certain purposes. So then how does that specificity arise? Obviously, it's not just carbon going down and going out into the soil as a carbon source. There are very specific signals that the plant sends out to signal to the bacteria around it to attract certain ones, to exclude other ones, to switch genes on inside the bacteria that make them perform certain functions. And in return, bacteria produce a whole lot of signals that are required for them to be recognized by the plant, to infect, and so on. And I want to talk about one of these signaling systems that we've discovered to occur between plants and bacteria that could be of very broad importance. So most bacteria are not really just acting as single-celled organisms. They interact with each other. And they do that by communicating. They use signaling molecules, so-called quorum-sensing signals, that they use to talk to each other. And they can use these signals to gauge population density, which then triggers certain behaviors. This is one class of these signals. They're called acyl homoserine lactones. They have this structure, and the side group is specific for certain bacteria that produce them. So different bacterial species produce very specific language signals, or mixtures of these signals. And production and perception of these signals is really important, for example, for biofilm formation, not just in plants, but for all bacteria, biofilm formation, synthesis of virulence factors that they might need to invade a host, infection of hosts, nitrogen fixation, and many others. So without these language signals, the bacteria cannot get themselves organized. They cannot switch into certain behaviors. So we thought, well, if plants have evolved over long times with bacteria, sure enough, they should have come across these signals. And so we've been looking at how plants actually interpret these signals from bacteria. And initially we used a technique called proteomics, where you just look at all the proteins that are altered in the plant in response to these signals. And we found plants react very, very specifically, and they're very sensitive to being exposed to these signals from the bacteria. So they can differentiate signals from different bacterial species, and also they can differentiate different concentrations that would reflect certain bacterial densities. And so maybe this could tell plants that certain bacteria are accumulating around them, and also whether they are actually ready to switch into a certain mode to turn on certain behaviors. So we think that plants have receptors to these signals, which are not identified yet. So they perceive these signals from the bacteria, and they have responses. And some of these responses are altered defense responses. So when they perceive that bacteria are accumulating, they modulate their defense responses against these bacteria locally in the root. But they can also send signals up to the shoot, for example, and warn the shoot. And this can, in some cases, make the shoot resistant to other pathogens that come and infect them later. And another interesting aspect that happens is that the plants actually decide to interfere with this communication. So they produce mimic signals that mimic the bacterial communication signals. And these mimic signals have been detected, not identified yet, detected in many plants. And they could basically, some of them work as agonists and some as antagonists of these bacterial quorum sensing signals. So the plant, there could be a group of bacteria building up around the plant. They might be plotting some kind of attack by communicating with their communication signals. But the plant can detect these signals and send out mimic signals that could then disturb that communication and basically stop certain behaviors, for example, biofilm formation. So do plants have an advantage from detecting these signals? We're trying to find out if we can warn the plants by letting them perceive these signals and altering their behavior towards microbes. And one thing that we can do, for example, is increase symbiotic interaction with rhizobia. So this is a legume that forms nodules in response to rhizobia, but if you give them a little whiff of these signals just beforehand, they actually enhance nodulation. And there are also other responses in the plant that change when they perceive these signals. So it could be that they have a warning that some interaction is going to happen, and they alter defense responses that can let these bacteria in. And another thing that actually happens in legumes is that they produce mimic signals that actually get the bacteria to start talking to each other rather than stopping it. So the plant actually produces signals that mimic quorum sensing signals, and switch on the communication of these cells. Here's an example of one of the group of compounds that we've identified that does this. This is a plant just under a fluorescent microscope. And if we let that grow on a lawn of bacteria that glows red when it perceives quorum sensing signals, you can see that something is exuded from the root that switches the bacterial communication on. It actually makes them talk. And here's a plant mutant that is defective in producing flavonoids, a class of compounds that has many functions in plants and plant microbe interactions. And this mutant cannot do this. So this class of compounds seems to be important in mimicking these bacterial communication signals. So we think that this could be of importance for interactions of plants with a whole lot of microbes. So the microbes are out in the soil, they produce quorum sensing signals, and they actually alter the exudation of signals from the plant, in this case flavonoid signals. These in turn get exuded into the soil and they act as mimic signals for the bacterial quorum sensing signals. So bacteria think that there are actually more of them around than there really are, and they start switching into a certain behavioral mode. So what could this lead to? By altering these signals, we could maybe alter signaling that could stop biofilm formation or enhance biofilm formation of symbionts, signals that enhance cooperation of bacterial cells, or signals maybe that stop pathogens from talking and carrying out certain behaviors. And I want to thank a whole lot of people, and these ones are just the people who have been involved in this project as well as the funding sources. Thank you.
Thank you very much. Questions or comments? Please don't forget to state your name and location.
Yes, Dick Manchester from CSIRO. So what physical form do these signals have?
The mimic signals, or?
Yeah, well, you talked a lot about signals going this way and that way. What, are they chemicals or are they?
They're chemical signals, yes. There are hundreds of signals really between roots and microbes. Some of them have been studied in detail. So in this case, the quorum sensing signals are acyl, homoserine, lactones. And the plant signals are flavonoids, so they're phenolic compounds. But plants exude hundreds of potential signals. Many of them we don't know what they do. But yeah, they're different chemicals. And usually it's a core structure of a chemical, and the specificity is achieved by certain side groups on that molecule. In both of these cases, the mimics and the quorum sensing signals.
Boris. Not Boris.
Boris Martinak from Victor Chang Cardiac Research Institute in Sydney. Question is, do all plants use these mimics to attract the nitrogen-fixing bacteria? Or if they do, which are doing better than the other? Or is there, my question is aiming at if this is one way of improving the growth of plants and so on, it might be a good idea to try to get all the plants which are somehow useful, giving the lack of oxygen, less and less oxygen on the planet and so on, to get them inserted a gene which will help them attract bacteria for nitrogen fixing. Yeah.
Yeah. So we don't really know if all plants do this. Flavonoids are specifically they are produced by all plants, but there might be very specific ones that switch on quorum sensing in bacteria, so we don't know yet exactly which ones they are. We are manipulating the flavonoid pathway to see what consequences that has on. It might have many other consequences. This is, once you get it into the field, there are lots of other things that happen to those signals in the soil. They get degraded or modified by bacteria, so it's actually quite complex.
I think in the interest of time we should move along, but please take the opportunity to ask questions at lunch.